Good day, this is the Professor, CBMD PhD. It's today Sunday, July 24th, 2022. It is 5:25 and 3 seconds, a.m. Eastern Standard Time. In today's research, embryos. So let's hop to it here. The study finds why many in vitro fertilization embryos fail to develop. Date July 19, 2022. Source, Columbia University. The spontaneous errors in the earliest phase of cell division may be the reason why so many human embryos fail to develop normally, according to new research. Approximately 24 hours after a human egg is fertilized, the process of cell division begins. During cell division, the entire genome, 46 chromosomes containing more than 3 billion base pairs of DNA, must be faithfully duplicated. The duplicate sets of chromosomes must then be separated so that each daughter cell receives a complete set. In many human embryos created for in vitro fertilization, something goes wrong, and some cells within the embryo have too few or too many chromosomes. Researchers have long theorized that errors occur during the final phase of cell division, when the duplicate sets of chromosomes separate into two identical daughter cells. Most of these failures were attributed to issues with the microtubule spindle, the apparatus that pulls the two sets of chromosomes apart. The studies found that chromosomal abnormalities stem from errors that occur much earlier in the process of cell division, when the genome's DNA is duplicated. And if the DNA is not copied precisely, his studies found the spindle malfunctions and places the wrong number of chromosomes into each daughter cell. When DNA duplication is abnormal, the spindle does not function normally, and this has largely been overlooked in previous studies. Because why would the embryo allow the integrity of the genome to be compromised when this is such a critical requirement for normal development? And though the studies were conducted with embryos created in a petri dish, including from individuals undergoing in vitro fertilization and egg donors who were not seeking fertility treatment, the same problems may contribute to the failure of embryos created in natural human reproduction. The source of DNA copying errors in embryos appears to spring from obstacles within the DNA's double helix, and though the precise reason for these obstacles is not yet known, they cause duplication of the DNA deposits, or even stop, which results in DNA breakage and an abnormal number of chromosomes. The spontaneous DNA errors can occur as early as the first cycle of cell division in human embryos, the researchers found, as well as in subsequent cell divisions. And if too many cells in the early embryo are affected by chromosomal abnormalities, the embryo cannot develop further. Most human embryos created for in vitro fertilization stop developing within days after fertilization, and this inefficiency of human development is an obstacle to successful fertility treatments. Many women undergoing fertility treatment require multiple in vitro fertilization cycles in order to get pregnant, and some never get pregnant at all. And not only is this enormously expensive, it is emotionally taxing. The researchers are planning additional studies looking at DNA damage during replication in the hope of understanding normal and disease causing variations in the human germ line. In the long term, these studies may lead to methods to reduce the risk of genetic abnormalities and embryo attrition for patients undergoing in vitro fertilization. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.